You know, every single cell in your body has this incredible blueprint inside it, right? Your DNA. It's the instruction manual for, well, for you. But actually being able to read that manual perfectly, that's been one of the biggest challenges in medicine. Today, we're going to dive into how AI is finally teaching us to read our own code with a kind of accuracy we've never seen before, and how it's literally changing lives.
So let's really think about this. Imagine your genome is the book of life, a book with 3 billion letters. Now, a single typo in that book, what scientists call a genetic variant, that can be the difference between being perfectly healthy and having a devastating disease. So the real problem in genomics has always been this. How in the world do you find that one critical typo, and, just as important, how do you find it fast enough to actually save a life?
All right, so if we're going to find these typos, first things first, you need a reliable way to actually read the book, right?
Well, that whole process has a name. It's called variant calling. Basically, we sequence a person's DNA, which chops it up into millions of tiny fragments. Then, we compare all those fragments to a standard reference genome to see what's different. And those differences, those are the variants. Finding them is absolutely crucial for everything, from diagnosing a rare genetic disease in a newborn to discovering the targets for brand new, life-saving drugs.
Yeah, but here's the thing. It's just not that simple. The sequencing machines themselves aren't perfect, and trying to align millions of those little DNA fragments in exactly the right place, it's incredibly hard. All of this introduces a ton of electronic and chemical noise. So what you end up with is this huge, messy signal versus noise problem. Is that little blip you're seeing a real disease-causing variant, or is it just a random glitch from the machine? Getting that call wrong can have massive consequences.
You see, the old-school statistical methods were pretty good, but they just kept hitting a wall with all this noise. So a team at Google came up with this really radical new idea. They asked, what if we stopped treating this like a statistics problem and started treating it like an image recognition problem?
And that's how we got a tool called Deep Variant. And the core idea here was just genius. Instead of feeding a model a bunch of raw numbers, they figured out how to turn the aligned DNA data at any given spot into an image. They call it a pile-up image. It's a picture that visually represents all the evidence. This meant they could use the same kind of powerful deep learning models that are great at, say, identifying cats in photos to learn how to spot genetic variants.
Okay, so take a look at this. It's actually pretty wild how it works. Think of it this way. Each row you see is one of those little DNA fragments we sequenced. The columns, that's all the different data about them, like the DNA letters themselves, how confident the machine was about that letter, how well it lined up with the reference, all of it. The AI learns to look at this whole picture all at once, just like a trained human expert would, to make a final call.
And this is a perfect example of what the AI is actually seeing. On the left, you've got a super clear-cut case. All the DNA reads agree, they all show the same variant. That's a classic homozygous variant. It means you got the same version from both your parents. Then on the right, it's a perfect 50-50 split. That's a heterozygous case, where you've got one version from the reference and one version that's the variant. Easy peasy for the model.
Okay, but what about all that noise we were talking about? Check this out. At first glance, a few of these reads look like they're showing a variant. But then, the AI looks at the other data channels, and it sees their quality scores are just awful. So it looks at the whole picture and correctly learns, nope, ignore those. That's a technical error, a glitch in the machine, not a real variant.
So, what's the bottom line here? The bottom line is, this crazy idea worked. And it didn't just work. It worked incredibly well. Deep Variant just blew past the accuracy of all the older methods. And it wasn't just more accurate. Its confidence scores were way better calibrated. That means when Deep Variant says it's 99% sure something is real, it's actually truly 99% likely to be real. This was a huge, huge leap forward for the field.
What's so cool is that this new AI approach wasn't just about solving an old problem. Nope. Its real power, its flexibility, was about to unlock a whole new generation of DNA sequencing tech.
So for a long time, the gold standard was something called short-read sequencing. It gives you really accurate but teeny tiny snippets of DNA. But then this new technology, long-read sequencing, came along, and it offered huge advantages. We're talking reads that are thousands of letters long. So they can easily cross these really difficult, repetitive parts of our genome, where short reads just get completely lost. But there was a catch. The early versions had a different and often higher error rate, especially for little insertions or deletions.
And this chart right here shows you exactly why that's such a big deal. In the easy parts of the genome, sure, both technologies do great. But in those really tough, repetitive neighborhoods, places like segmental duplications, the accuracy of short-read methods just plummets. But the long reads, because they can see the whole context, they stay incredibly accurate. They were the key to finally seeing the full picture.
And this is where the AI approach is just a total game changer. Instead of having to build a completely new statistical model from the ground up to handle long reads, the researchers could just retrain deep variant. They just fed it a bunch of examples from new long-read sequencers, like those from PacBio, and the model simply learned the new error patterns. It just adapted. This proved that deep learning could give you all the incredible strengths of long-read sequencing without any of the downsides.
So, okay, that's the tech. But what does this powerful combo of long reads and a super flexible AI get you in the real world? It gets you this: less than 8 hours.
And this is the system that pulled it off. A team of researchers at Stanford and UC Santa Cruz built this amazing pipeline. They used long-read Oxford nanopore sequencers and a specialized workflow that included deep variant, and they were able to go from a patient's blood sample to a final genetic diagnosis in under 8 hours. That's a world record. And in a critical care unit, that's not just a number. That can be the difference between life and death.
Okay, so we're getting incredibly good at reading one person's genome, right? Fast, accurate. But all that progress shined a massive spotlight on a much, much bigger problem. Whose genome are we actually comparing it to? You see, for decades, the standard reference genome, the so-called normal we compare everyone against, was based on just a tiny handful of people, most of them with European ancestry. And as the Human Pangenome Reference Consortium puts it right here, that single reference is just insufficient to capture the extent of sequence diversity. This creates a huge problem of equity, where our best medical tools literally work better for some people than for others.
The solution to this huge problem? It's called the pangenome. Instead of one flat, linear book, think of the pangenome as this incredible, dynamic map that includes the genetic diversity from hundreds of people from all over the world. It is a much more complete and much more equitable book of life that actually starts to look like all of us.
And this is where it all comes together. This is where those threads we've been following, the long reads, the smarter AI, and this better, more equitable reference, they all weave together into this amazing, powerful cycle of innovation.
Here's how it works. Step 1, you get these ultra-accurate long reads, like from PacBio's Hi-Fi technology. They're like the premium building blocks. Step 2, another AI tool, Deep Consensus, comes in and polishes those reads, making them even more accurate. Think of it as creating even better building materials. Step 3, that super high-quality data lets projects like the Human Pangenome Consortium build their amazing diverse reference maps. And finally, Step 4, by combining deep variant with this new, richer pangenome reference, you get more accurate and more equitable diagnoses for absolutely everyone. Each innovation just fuels the next one.
I mean, think about it. We've gone from squinting at our genetic code, struggling to find a single typo, to now being able to read this vast, diverse library that represents all of humanity, with AI as our guide. It just leaves you with this one massive question. Now that we have a more complete map of who we are and the AI to read it, what are we going to discover next? What diseases are we finally going to be able to diagnose, treat, or maybe even cure?